Hello, my name is Phil Withers. I'm a senior lecturer in paramedic science at the University of Hertfordshire. Welcome to this taster session on cardiac electrophysiology and the electrocardiogram. We've put this taster session together for you just to give you a little bit of an insight of our style of lecture at Hertfordshire, and it starts to introduce you into some of the clinical aspects of our role as paramedics. So to start with, you may have seen an ECG machine before, an electrocardiogram, and it has a very wide application in the clinical world. Most modern ambulances and paramedic response cars carry portable ECG machines, and we use them for a variety of patient presentations. So most common being chest pain when we're looking for signs of heart attack. If a patient has collapsed and we want to see the activity in their heart. Maybe shortness of breath, which could be a side effect of a cardiac problem. Dizziness, if the heart isn't beating as it should and perfusing the brain with oxygen, the patient may feel dizzy. A patient may be complaining of palpitations, which is the sensation of their heart beating in their chest. It may be an arrhythmia, which we can pick up on an ECG machine. And of course, we use it to monitor a patient's heartbeat while they're in our care. And the good thing about modern ECG machines, they're quick to apply these electrodes that we stick onto the patient's chest. It's non-invasive, we don't break the skin, it's painless, and it's diagnostic for us. We can diagnose a number of conditions through a very rapid ECG. Now, in order to understand what an ECG produces for us, we've got to have some knowledge of cardiac anatomy and physiology, and that's what we're going to go through on this session. So the heart, as you probably know, is a pumping organ, and it's a double pump because it pumps blood both to the body, which we call the systemic circulation, but also it pumps blood to the lungs as well, which we call the pulmonary circulation. And the heart is divided into left and right sides. So the left side ejects blood into the aorta, which is your largest artery, and that takes blood around the body. And then when blood comes back to the heart, it goes into the right side of the heart. And from the right side of the heart, it's ejected into the pulmonary arteries, and that goes off to the lungs for the blood to be refilled with oxygen. And then it comes back to the left side of the heart and out to the body again. So if we cut through the heart, we can see its 4 pumping chambers. So the left and the right atria, that's where the blood comes into the heart, and the left and the right ventricles, and those are the big pumping chambers that eject blood. And you can see on this diagram the walls of the left ventricle are much thicker than that of the right, because the left side has got to create enough pressure for that blood to go around the whole body, and the right side less so because it's only going to the lungs, which are next door to the heart. Now, in order for the heart to be successful in pumping blood at the right time, there needs to be some sort of coordination and control of the pumping action. And that control gives us what's known as the cardiac cycle. So we get these periods of contraction and periods of relaxation. And the periods of contraction we refer to as systole, and the periods of relaxation we refer to as diastole. You can also see on this diagram the structures that stop blood going backwards through the heart, and they're known as the heart valves. So we have valves that are between the atria and the ventricles, known as the atrioventricular valves, and we have the semilunar valves that are between the ventricles and the arteries that leave the heart. So the aorta from the left side and the pulmonary artery from the right side. And those valves stop blood going backwards. Now, of course, these can go wrong, but that's a whole other subject. So in order for the heart to produce enough force, it needs this coordinated control. The pumping needs to be efficient, it needs to be regular, and it needs to be coordinated as well. We can't have different parts of the heart contracting at different times. And the heart is made up of cells, which we'll find out more about in a moment, but it's important that these cells, these muscle cells, contract at the right time in order for the heart to do its job. And we have two types of cell in the heart muscle. The cardiomyocytes, which form the majority of the heart structure, but also pacemaker cells, and these are key for timing of contraction. So if we were to take a cut through of the wall of the heart, we'll find a number of layers. On the outside, we have a tough and protective layer known as the pericardium. The thickest part is known as the myocardium. Myo meaning muscle. That's the thick muscular wall of the heart, which does the majority of the contraction to eject blood. And finally, we have the endocardium, which is the inner layer, which is very smooth, and that's what our blood is against inside the heart. So if we were to take a much closer look at the myocardium under a microscope, we'll find the cells, and these are the cardiomyocytes. Unlike other cells, they have a nucleus, and they have mitochondria as well, and you probably know that mitochondria are referred to as the powerhouses of the cell. They produce a substance called ATP, adenosine triphosphate, which which is the energy molecule. And there are huge numbers of mitochondria in cardiomyocytes because these cardiomyocytes start working as soon as the heart starts to form as an embryo, right through to the day you die. And they're working all the time. So their energy demand is huge. The cardiomyocytes, as you can see, are up against their neighbouring cells, so the cardiomyocytes that are next door. And the connection between cardiomyocytes is very specialised. This isn't found much anywhere else in the body. And the cardiomyocytes have an ability to communicate with each other via something called a gap junction. So when one cardiomyocyte gets the impulse to contract, it then sends that impulse to its neighbour. You may have heard of synapses in nerves. The gap junction is a specialist type of synapse known as an electrical synapse. So its message is sent to the cell next to it. Because of the amount of force that these cardiomyocytes generate, they do pull against each other. So they are held together by a structure known as an intercalated disc. This holds the cardiomyocytes together and stops them tearing apart from each other. Cardiomyocytes are very similar to your skeletal muscle, so the muscle responsible for moving your body around. But they are shorter and less circular in the transverse section than skeletal muscle. But they are made up still of these myofibrils known as actin and myosin, and they slide across each other and thereby shortening the length of the cell, and thereby when you add all these together, the thousands and thousands that go to make up the heart, they pull together, reduce the volume of those chambers, and thereby eject blood. And in order to get this to happen, we need ATP, which is that energy molecule, and we need the cation calcium ion in order for that contraction to occur. And if we were to look at a cardiomyocyte under a microscope, this is what we'd see. We can see on this diagram on the right, we can see those striations, those layers of cells, and them being held together. We can see the nucleus, which is highlighted in purple. And so when these contract, they produce the overall effect of ejecting blood. But how does this occur? What else is required for this calcium to get into the cell? Well, the impulse for a cardiomyocyte to contract comes from its neighbor. And as soon as it receives that impulse, it then contracts itself. So we get this wave. The important thing to know about these muscle cells is that their voltage changes in order for it to contract. So at rest, a cardiomyocyte has a charge of about minus 90 millivolts. As soon as the cardiomyocyte next to it sends its signal through a gap junction, sodium rushes into the cell, and the sodium ion being positively charged, it thereby changes the cell from being negatively charged to being positively charged. This is known as depolarization, and this is the very basis of an ECG machine. So you get this changing from a negative charge to a positive charge. Once it becomes positively charged, this causes calcium to come into the cell as well, and that's the calcium that's used, ultimately used, in order for the cardiomyocyte to contract. We get this plateau phase where the contraction is, and then the cardiomyocyte needs to return to its resting state. So it needs to go from a positively charged state to a negatively charged state, and this is known as repolarization. And it's depolarization and repolarization that we can see on an ECG. Now, I mentioned earlier that cardiomyocytes make up most of the heart muscle, but we also have specialized fibers known as autorhythmic fibers that are responsible for generating the heartbeat. So we know that the signal gets passed from one to the other, but where does it all start? Well, this is our conduction system. We have a natural pacemaker in the heart that spontaneously depolarizes, so it doesn't rely upon a signal from the cells next to it. It does it automatically. So this spontaneous depolarization of the pacemaker cells initiates the heartbeat. And here's an image of the conduction system. You may know that the SA node, the sinoatrial node, is the heart's natural pacemaker. This depolarizes spontaneously, and then that wave of depolarization spreads through the conduction system and through the myocardium, producing a coordinated contraction. So let's look at the ECG. Now we know a little bit about the anatomy and the physiology. How can we use an ECG to look at what the heart is doing? Well, this was one of the first ECG machines. The guy in the photo there is a chap called Wilhelm Einthoven. Invented the first ECG machine in about 1895. And you can see here that he's got his limbs immersed in buckets of solution, and it's actually salt water containing ions, because we are measuring those electrical changes in the cardiomyocytes, and that those electrical signals can be detected on the surface of the skin. Now, of course, things have moved on a bit, and this is a more modern electrocardiogram with sticky electrodes applied to the patient's skin. And whenever we stick an electrode on the patient's skin, we're able to see a waveform. And these waveforms are generated by depolarization, that changing of the cardiomyocyte from negative to a positive state. Now, of course, there are thousands and thousands of cardiomyocytes, so the ECG takes an average, if you like, of the direction. When we stick our electrodes on, we get these waveforms, and the waveform might be positive, so it's pointing upwards, so this one that we can see on the right side of the screen. If the wave of depolarization is moving towards that electrode, we get a positive wave, and if it's moving away from it, as we see on the left side, we get a negative wave. So this explains those ups and downs that you might have seen on an ECG waveform. We can break the waveform down into a number of waves and segments, and these correspond with the depolarization that are happening in the different parts of the heart. So the P wave represents the depolarization of the atria, the top two chambers of the heart. The QRS complex is made of the Q wave, the R wave and the S wave, and this represents depolarization of the ventricles, so the largest part of the heart, very tall wave because of the large amount of myocardium. So to begin with, the septum depolarizes, so that's the wall separating the left and the right side of the heart. Then we get the main mass of the heart, and then near the base of the heart, so as it returns. And then finally the T wave, well, this represents repolarization. So you remember that those cardiomyocytes have to change back from being positive to negative via the potassium ions leaving the cell, and that's what the T wave represents. Now, we've mentioned electrodes. Well, we can stick electrodes in a number of places to get different views of the heart, so we can look at different aspects. So when we use an ECG for diagnostic purposes, we undertake what's known as a 12-lead ECG. And lead in this case means a view, so we get 12 views of the heart that are useful for us diagnostically. So we stick electrodes on the limbs, which are known as limb leads, and we stick electrodes on the chest, known as chest or precordial leads. And in a normal, healthy individual, this is what we see. And we can see our 12 views here. So then they are 1, 2, and 3, which are down the left side, AVR, AVL, and AVF, and then V1, V2, V3, V4, V5, V6, and they are our 12 views of the heart. And the waveforms all look slightly different because we're getting different views of the heart, and this depolarization is happening in different directions relative to the electrode. Along the bottom you can see a lead 2. This one we use predominantly for monitoring. So why are ECGs useful for paramedics? Well, the one example I'm going to give you is this ECG. So if we look at particularly lead 2, lead 3 and lead AVF, we can see that looks very different to the previous slide. Something very odd is happening there between the QRS complex and the T wave. And also if we look in 1 and AVL, again we've got a very strange relationship between the QRS complex and the T wave. And this patient is having a heart attack. They're having what's known as a myocardial infarction. So their myocardium is starting to infarct, starting to die, commonly known as a heart attack. So this is one application of our ECG acquisition and interpretation, is to spot these patients and then treat them with pain relief, with medications that reduce platelet aggregation. We may give them a drug called glycerin trinitrate, GTN, to reduce the workload of the heart, to take the stress off it, and perhaps oxygen if the patient needs it. And then we'll convey them to a specialist hospital so they can have this problem fixed. So I hope that was useful for you, just a little overview of cardiac anatomy, some physiology, and then relating it to the ECG machine that we use extensively in our clinical practice as paramedics.